right. Welcome, everybody. Thank you all for joining our next webinar. Today's speaker, we're super stoked, is Dr. Gareth Trubl, or do I say it Trubl? How do I
It's
pronounce
Trubl.
it? Trubl, not trouble, right? Who is a soil microbial ecologist in the Division of Biosciences and Biotechnology at Lawrence Livermore National Laboratory. And he received his BS in environmental sciences with an emphasis on microbiology at the University of Arizona, and then followed that up with a master's in environmental sciences from the University of Nevada, Reno, where he actually worked at the Desert Research Institute looking at Antarctica, at Lake Vida Brine in Antarctica, which is really fun. Hopefully you've got some cool pictures of that in your talk too. Followed that by that, he earned his PhD in microbiology from the Ohio State University, where he's been really pioneering a lot of work in soil viromics. And he's joined the LNL since 2019. He's been working on a variety of sort of meta-omic approaches for isotope probing and detecting and characterizing viruses, virus-microbe interactions in soil ecosystems. So thank you so much for joining us. And with that, I will let you take it away.
Okay, yeah, thank you so much. So yeah, unfortunately, I won't have any Antarctic slides. They are cool. I do recommend people looking out, especially at Lake Vida for the cool microbiology. So I kind of have my talk structured for people who may not know about viruses, because I want to keep everyone engaged. So we'll start off talking a little bit about, what viruses are, how we look at viruses. The main tools we use is largely metagenomics, kind of what we've learned from marine environments where they've explored virus ecology for a really long time, and then moving into soil and kind of highlighting some of the projects I'm currently working on. Starting off first, let's see if this is going to work. There we go. If we think about what a virus is, there's a lot of semantics out there. Are viruses alive or not? And da da da. I would argue that it doesn't really matter, because the idea is if we know there's a virus, then we know there's life. Viruses are acellular biological entities. They don't have a general composition like microbes do, right? They are nucleic acid with a protein capsid and possibly a tail, especially if they're bacteriophage. So there's no cell membrane, there's no nucleus, and really there's no ATP generation. What's important about them is that they are so abundant. So this is a figure I've adapted here from Aristotle, where you can see in yellow, we kind of have prokaryotes, kind of a bad word, bacteria and archaea. We have eukaryotes, which is everything else that's, multicellular. And then we have viruses, and they're sheer abundance. There's so many of them. And viruses can infect everything, right? They can infect humans, they can infect plants, fungi, they can infect bacteria, which is largely what I study. And the bottom left, I actually have viruses infecting giant viruses. So even viruses can be infected by other viruses. So we typically think of a virus as something that's so small and so hard to work with. And that's largely true, but we need to think about scale. So on the left here, I have a bacterium, E. coli. That's kind of like our model we always think about. Everything else on the slide is a virus. And I have the names color coded by what they infect. So in the bottom right, we have our human viruses. A lot of, medical students care about that and focus on this. So I have you recognize there. We have these large amoeba viruses that we've found for the past 2 decades now, in this purple color. And then we have archaeoviruses. And then the bottom left, we have bacteriophage. And these are just a couple examples. And this is largely what we see when we're looking at ecosystems. And a large part of that could just be because of the bias of our methods. So viruses have two main replication strategies or infection styles. The first is if you think about a virulent virus, or this is a obligately lytic or professionally lytic virus. This is what you think of when you think of a virus. It comes into a cell, and here I have a bacteriophage, but it's not always bacteriophage. But for a bacteriophage here, it's injecting its nucleic acid. It takes over the host's metabolism, tells the host, you need to copy more of my nucleic acid, and then you need to make my proteins, and then they assemble into these progeny viruses, and then they burst, leaving the host. All right, so that is all in all what a virus wants to do, make more viruses. They can also be temperate, right? And this is where they do lytic infection, but they can also do lysogenic infection. So on the bottom here, the lysogenic infections where they insert their nucleic acid. Nucleic acid can remain in cytoplasm. It can be part of a plasmid. It can be integrated into the host chromosome, and then it gets copied with the host. Now, I want to argue that this doesn't mean the virus is dormant. This is not the same thing as a remnant, right? This is a whole virus that's in the host that's replicating with the host, and it is doing things. And this can be a wide range of things. But essentially, every time the host doubles, the virus gets doubled as well. Then there's also a small set of viruses. We call these chronic infection. They're part of this lysogenic cycle where they remain in the host, but then they can send out some virions here and there. And you can think of these as scouts. There may be other reasons for them. This is something that's still going on, but they can send a couple of viruses out into the environment. So how do we largely look? How do we even know this? And well, in the past, we've isolated viruses. And to isolate a virus, you have to isolate the host. So when you think about isolation, it can be really hard to isolate a virus because in order to see it, you need to do lytic infection. You need to grow a bacterium or a fungus, and you really need to see these plaques. You need to see this death to know, oh, there could be viruses there. But not all viruses do that, right? We said they can be temperate and some of them can be incorporated in the host as well. And so it just makes it really hard to culture viruses, especially because it's already hard to culture the host. So what's really taken off viral ecology in the last 2 decades has been metagenomics. And when we think of metagenomics, it is that we're looking at all of the DNA in a sample. So this is biased because when we were originally doing this, we were wanting to look at human DNA. So we're really all of our methods and everything have been really pushing for double-stranded DNA because it was based off originally looking for humans. So we largely see double-stranded DNA viruses. And in this figure, on the left, we have the number of sequences. This is a figure that's adapted, by the way. It's not going to look the same if you go back to the publication. But I love the background and what it actually means is pretty much the same. On the x-axis is a timeline of how things have changed over time. And really the thing to take out here is this, if you look at this yellow, in the last 2 decades, it's just exploded. The number of viral sequences that we can get from just sampling all of the DNA in the environment is the main way that we use to detect viruses. And we further expanded from this. So we can have metatranscriptomes to look at RNA viruses. Something I've helped pioneer as well is this idea of a virome, which you are pulling viruses from an environment and then just extracting nucleic acid from things of virus size. And I'll talk more about that later on. But when we've done these metagenomic studies, we've really seen that viruses are much more important than we thought they were. And we first learned this in the ocean. So what I'm going to lead you into is we found out these three dominant roles that viruses have in the environment. And there's many nuances to this and many different little differences here and there. But really, this was summed up in Meyer Breitbart's 2012 paper, Marine Viruses, Truth or Dare. And I really want to give a shout out because it's such a great paper. And it is the paper that I read that I thought, okay, viruses are important for ecology, which is what I care about. And this is when I got on board with viruses are important and I want to study them. So the first thing to think about is viruses here as a grim reaper, right? So in the oceans, when they were looking at data, and this is largely bacteria, that they were seeing that 1/3 of these bacterial cells were lysed every single day in the ocean. All right, so this generates about 10 billion tons of carbon every day. And for somebody who cares about the environment and thinking about climate change, that's huge. This is a huge role part of carbon cycling. Now, if you think about the virus, it's like, okay, it kills the host, there's more viruses. But let's think about what just died here. This bacterium, this microbial cell, right? Its guts, its innards are now out in the environment. So here's a transmission electron micrograph from Dr. Jennifer Brum. You can see here we have a bacterium that's infected, more virions are being produced, and eventually it lyses the host. And you can see all of that juicy nacromass. That's the guts, the innards, right? This is the nutrients. This is important stuff. This is organic matter. So there's two ways to really think about this. And the original way we thought is it's feeding other microbes. So viruses promote diversity. When they kill these most abundant microbes, they're kind of feeding everyone else. You can think of it kind of as a Robin Hood with nutrients, and they're feeding all these other microbes. But there's a paper that came out in 2016 by Guidi and others showed when they actually weren't even just looking at viruses, but they saw that when this necromass is being produced, viruses are aggregating, they're sticky, and it's actually causing the carbon to sink. So viruses are actually the best predictor of export of carbon to the deep ocean. So instead of just promoting diversity, it could be that maybe viruses are sequestering carbon too. It's going to the deep ocean where it's going to hang out for a bit. Now, their second role is that they're kind of drug dealers, they're smugglers, they move stuff around. And we've known this for a long time, and it's becoming more recently recognized how much viruses promote diversity, not just from killing hosts, but actually moving genetic material around. So there's a neat paper that kind of shows this, and I've kind of clipped two snippets here to kind of condense it. But really, it's like we thought about before via the two infection cycles. You have a virus that makes more viruses, it takes a little bit of the host DNA with it, or if you want to think about RNA as well, and then it takes on to the next host. So it's kind of packaging material and moving around in the environment, right? The other is when it's being replicated in the host over time, you can have viruses that get degraded, and this is how we get remnants that are kind of just hanging out. But they can get more and more host DNA in them. So when they go into a new host, they can actually deliver more host DNA. So viruses promote diversity not only by killing, but really moving material around. And I think the third thing is what has caught a lot of people's eyes is that viruses are wizards, they're the control all. And this is kind of where I'll argue it doesn't matter whether viruses are alive or not. We have a term for this, especially for single cells, where when a virus infects the host, it completely changes its behavior. It does not act like itself. So we'll call this a virus cell. And this is because the virus is in control now. This is not a bacterium that functions like normal. So you could arguably say the virus is alive in this state. So what happens is the virus will infect the host. Typically, it says, you know what? I'm going to stop you from doing all this other cool stuff. I really just want you to generate energy because I want to make more progeny viruses. So copy my nucleic acid, copy my proteins that come together. I'll leave you abandoned ship. That's cool. But that's not just all that happens. Sometimes environments can be harsh and there can be competition, and maybe there's not enough resources. So you need to beef up your host before you abandon ship. Before you jump ship, you want to get it closer to land. So here's where we actually have other ways you can reprogram the host. And one facet of that is really looking at these auxiliary metabolic genes, but it's not just about these AMGs. So these are genes that are host derived, so from a previous host, that have an additional function besides just virus replication and making progeny viruses. And I guess a great example is in the oceans where you have cyanophage that infect cyanobacteria like Prochlorococcus. And in this example, let's say the virus is infecting the bacterium. It wants to make more viruses, but it requires these nutrients and they're just maybe not available. So the virus encodes a gene, maybe it's PSBA or PSBD, which is for photosystem 2, which is the rate limiting step in photosynthesis. So it can actually make the host express this gene. The host effectively does photosynthesizes more efficiently, generating energy, and then it can make progeny viruses and leave the host, and now the host is dead. But during that period, it's not only changed the behavior, but it's actually produced more oxygen for us and taken up more CO2. So this is just other ways that viruses can indirectly impact the ecosystem and the global nutrient cycles. Moving on from here, now that I've given you kind of what is a virus, metagenomics is the main way we look at viruses. The oceans kind of laid the foundation of what we understand about viruses today. I'm moving into soils and how they're they're vastly understudied because they're super hard to work with, but it's well worth it. So I care about soils largely because they have so many ecosystem benefits, but they store a lot of carbon. They are the largest storage of carbon out of any terrestrial ecosystem. So they have as much carbon as all the biomass on Earth and all of the oxygen in the atmosphere combined. So this map of the world here shows if you see darker brown, it's where we think there's more carbon being stored. So you can see disproportionately there's more carbon along the equator and then in the northern latitude by the Arctic. So a lot of my research in the past has been in these Arctic ecosystems. And to really understand how much carbon there's 1,500 to about 24 gigatons of carbon. And that's a huge number, and we have a hard time comprehending huge numbers. If we think about it as you take a cow, we have so many cows now because everyone loves beef. But if you think of a whole cow as just carbon, nothing else, let's say it's about 1,500 pounds of just carbon. That would mean to equal the amount of carbon we have in the world, there's about 2 to 3.2 trillion cows in our soils, just made up of carbon. So there's a lot of carbon there. This carbon is important because it can be released as carbon dioxide, which is a major it's a greenhouse gas, which can increase global warming. But additionally, we can have it released as methane too, which is more potent, and that actually breaks down into CO2. So that's not good either. So my focus originally in environmental microbiology was I cared about environmental science. I took microbiology and realized microbes are major players and viruses control microbes. So I need to understand microbiology, right? Because microbes and viruses are important for climate change. Okay, so setting out, I was like, okay, we need to study viruses from soils, but soils are super complex and it largely hadn't been done before because they're so complex. And the idea here is if you think about how most of our information has come from isolates and metagenomes. Well, if you look at a typical soil metagenome, assembly is bad. It's really hard to work with it. There's a lot of contamination. There's so much diversity. Soils are the most diverse ecosystems on the planet. So they're just really hard. And if we just look at this figure here, a centimeter squared area, we have a lot going on, right? Just from what we can see, we're like, okay, we got plant roots, we have amoebas, we have fungi, there's a lot. And if we zoom in on just one pore space, we go from these larger things that we can see down to our microbes and our viruses. So just here, then we see kind of this biofilm or these bacteria that are together in this pore water. And then zooming in even more, we can now see the viruses. So in order to look at these viruses, especially from metagenome, think about that. We have to remove everything else from that sample. That is incredibly difficult. So our main tool, which is metagenomes, is really hard in soils because you can't isolate viruses. It's very difficult. You can, it's just difficult. And then when you do metagenomes, you are swamped by all this other material. So it's just low resolution on our viruses, right? So I largely spent my PhD figuring out how we can look at viruses in different ways. And largely this started with the virome. And I'm going to talk about that towards the end, but I want to talk more about what I've done more recently. And this is in order to get more resolution on viruses, we've been using tools to try to look at the ones that are specific for substrates. And so the way this comes in is with stable isotope probing. And I included this picture here because it's just a beautiful piece of art to just illustrates all the various applications we can have from stable isotope probing. And I know that's a lot, so I'm going to break it down. But largely, we can take different isotopes of substrates and track it through an environment. And so we use this in a form of 15N or 18O or 13C to track these nutrients in an environment or look at activity. And this can be used for any system, whether it's humans or whether it's environment, plants, you name it. So to break it down more, what a stable isotope is, I have two examples because I'm going to kind of be talking about these a little bit. On the left side, we have just water, and it's typically H2 and then 16O. So we have 8 protons, 8 neutrons. You can think of it as like a Tootsie Roll. And then we can make an isotopically labeled one where there's 2 more neutrons on the oxygen. So think of it as a flavored Tootsie Roll. So this doesn't affect biology in a negative way, as these 2 neutrons that don't really interact and they just add more mass there, right? On the right side, we have an example of what we do for carbon dioxide, where you can actually 13C label the carbon. And these are stable isotopes, so when we introduce them, they don't interact and they don't negatively affect the organism. So what we typically do is, and I have here regular water and our heavy water, is we'll take this substrate and then rich substrate, we'll add it to an environment. Here, I'll add it to a soil sample. We'll incubate it for a couple days. And if we specifically are using water, water gets incorporated into the backbone for all DNA that's made. So this means that we can actually track DNA synthesis. So everything that's active will make new DNA, and this DNA will become labeled. So we can actually look at who's active and who's not active in the ecosystem to increase our resolution by just focusing on the active one. And now that we have these organisms growing, the ones that are growing are uptaking the heavy water, so they're becoming labeled. We then spin it super fast via ultracentrifugation. The DNA that's in there gets separated based on its density. So the DNA that incorporated the heavy water is more dense, so it moves down in the tube, and the DNA that doesn't have it is lighter and up towards the top. So then we can extract these fractions and we sequence the non-labeled ones just to see who else is there and to have our control. And then we can start looking at active versus inactive populations. Okay, so first example of this is I like to do a lot of work in the Arctic. So my first project that I was doing this is in Bonanza Creek. This is up in Alaska, where we have soils that have permafrost, which is ground that's frozen for 2 or more consecutive years. And we know that permafrost is thawing. And so a big question is, what's going on with these landscapes? They're typically a sink for carbon. Are they going to become a source for carbon? And a major outstanding question was during the winter, when everything is just completely frozen and it's super cold, is anything active? Well, when we look in the soil, these are peatlands, we'll have these bogs. And bogs are typically inundated or partially inundated where they have some water in there, right? And the plant material, the sphagnum moss, gives off these exudates and these solutes that actually prevent the water from freezing. So we have this water that's sub-freezing below snow and ice potentially, but the it's still liquid state, so we could still possibly have activity. So we wanted to see, is anything active and is it emitting carbon dioxide over the winter? So we took these cores, we incubated them for a half a year and a full year with and without heavy water. We ultracentrifuged them. We took the heavy DNA to look at if anything was active. And what we found were a ton of viruses and only a few bacteria that were active. So in this figure I have here on the left, I have all these pill-looking shaped things. Those are the different bacterial populations that we identified. And there's ones that are active and that are larger, and there's ones that are inactive. And then if we look at the viruses, they are the ones that are hexagons, and the big ones that have numbers are the active viruses, and the ones that are smaller are the inactive viruses. So when you typically take a metagenome of this environment, all you see is that in pink, the actinobacteria, they dominate. They're the most abundant organism there. So if I just surveyed the soil, I would say actinobacteria are really important because they're highly abundant. But from our samples for a year incubation, we don't see that. We actually see that in green and yellow, our Bacteroidetes and our firmicutes, those are actually our abundant bacteria that are active. So by using stabilized probing, we can actually get resolution and see who is actually contributing to these ecosystems. And we see the same signal with the viruses, that while these are the active ones, they actually have the most viruses infecting them. And that when we actually look at this relationship between viral infection and then production, we were measuring CO2 during this period, and we saw that indeed there was carbon dioxide being emitted from these soils over the winter months. So it's not like we thought before, where when it's really cold, a lot of stuff is frozen, there's no carbon dioxide being produced. There is carbon leaving the soils. So it's something important to consider when we're modeling carbon leaving our soils. Okay, so moving on to our next project. This is one that we're still working on. This is going to northern Sweden, just above the Arctic Circle. We have another peatland, but this is different. Instead of just having a lot of bog, which is the sphagnum moss, which you see here in the middle, this is actually a natural permafrost thaw gradient where we can actually look at intact permafrost, which is pulsa, partially thawed permafrost, which is our bog, and then our fully thawed permafrost, which is Sven. And this very much resembled a wetland environment that you would see going outdoors, right? Where you have standing water. And this is typically measured with high methane emissions. So here, what we did, instead of using stable isotopes, we worked on extracting all of the viruses and things of virus size from the soil. So the first thing I did was I used methods that we pioneered to really shake and desorb viruses from the soil matrix, right? So we resuspended these virus particles and also things of virus size. There are ultra small bacteria and archaea in there as well. And then we do a size filtration to isolate them, and we extract just the nucleic acid. So here we were looking at double and single stranded DNA viruses and things of virus size. And then we did a whole range of bioinformatics to look at their ecology by doing a global comparison, looking at their AMGs, and then looking to their host or linking them to their host to really see their impact overall on the environment. And the major takeaway that I'll distill it down to is similar to what we've seen before, but now with kind of more research, is that viruses in soils, especially these soils that have a lot of plant material, they have a lot of these auxiliary metabolic genes that help break down organic matter. So a great example is from a previous paper we published led by Joanne Emerson, where we see these peatlands have these complex carbs, right? These are these large polysaccharides, really hard to break down. And the best way to think about it is if you're trying to eat a pineapple with the rind, you're going to do it if you're hungry, but it's going to suck. You're not going to be happy. So these do get broken down. It's just very slow and takes time. So the viruses encode these glycoside hydrolases, which break specific bonds in these polysaccharides, turning them into, or when having the host express it, so the host digests it and breaks it down into these monomers. So these monomers are easily digestible by other organisms. So now instead of that pineapple, you have either a burger or if you're vegetarian, you have a salad, something that you enjoy, you can eat rapidly, and you like it, right? So now that we have these simple carbs that are being broken down by all these organisms, what does this mean for carbon? Well, it means that instead of this carbon just hanging out and being slowly degraded, it's now more easily degraded and it's being released as carbon dioxide and as methane from these environments. So in this situation, it could be that, viruses are helping promote diversity, feeding everything, helping everything eat. But then now there's more organisms and more metabolisms that are actually releasing carbon from the soil. So we don't know if that's the whole story, but that's kind of what our findings are looking at. Okay. Now, moving in kind of to our next area, and this is kind of where my research is going right now, where we are looking at a completely different system. Before I was talking about the Arctic and these really more not limited, but not as diverse ecosystems. But now we have these grasslands, right? And in here, this is largely controlled by water. So we're using water as a master controller. And if anyone's ever been to California, you know that there's just not a lot of rain during the summer. Typically from May to September, we just don't get rainfall. And in Southern California, you don't have as many grasses as you do in Northern California, but everything just kind of turns yellow and brown, and it's just kind of persisting. And then after you have your first rainfall, everything starts to come alive. Well, this first rainfall is associated with a giant release of CO2 from the soil, so kind of like a belch, and this is called the Birch effect, right? So the soil leaving, or carbon dioxide leaving the soil. So I'm part of this larger science focus area funded by the Department of Energy. We're really trying to understand how water is a master controller of these microbial and viral populations, looking at this when there's a lot of rain around, and then also when it's drought, and we're looking at how microbes and viruses are persisting and how this affects their activity and their overall traits. So we do this with heavy water to look activity. We also, our field sites in Northern California, about 2 hours north of Livermore, an hour north of San Francisco, where we have chambers where we can actually grow grass that's 13C labeled to really track interactions with the soil microbiota. So from this, my portion was really looking at the viruses. And something that we saw before is that the microbes there, how they die, specifically bacteria, really matters. So I wanted to conduct an experiment looking at how something dies and specifically looking at viral lysis, because that's what's important to me. But just to give you some larger context of the flavors of other work that's going on, there's this review we put out in 2022, where if you look on the top right, you have this bacterium. And we really looked at how the way it died influenced the ecosystem. So top left, we have osmotic shock. This could be when there's a period of drought and then the rapid freshwater influx from rainfall. This could be bacterial predation. We have other bacteria that kind of act like vampires and they attach to other bacteria and drain their nutrients. We have grazing from kind of these protists. You have chemical warfare from bacteria and fungi fighting with each other and producing these antimicrobials. You have desiccation just when you have this drought and there's just no water and it shrivels up. And then you have viral lysis. And really, each way the bacterium dies affects its nutrients, the necromass, right? If you think of it as being desiccated, well, you just have this dry shell, right? You still have your cell membrane, and it just has kind of these dried up nutrients. It's harder to use. Chemical warfare, you want to go use something, but then you potentially have something that's going to affect you as well. Well, likewise, with viral lysis, there's been previous papers showing that, viruses are not the same as their hosts. So when they make progeny viruses, they don't actually use all the host nutrients, but they use a lot of it. And they actually disproportionately use more phosphorus compared to carbon and nitrogen because they're making viruses which don't have all the fatty acids and stuff that microbes do. So this got me thinking, well, maybe viruses are limiting productivity in soils because nitrogen can be limited, phosphorus can be limited, and if viruses are disproportionately taking phosphorus out of the system, and these virions are persisting, maybe they are the secret storage of phosphorus for soil ecosystems. So I took soils from Hopland that I showed you before that were dry from that summer, and then I added heavy water or regular water with and without phosphorus. And then I sampled them right before adding water as dry soil. And I sampled 1, 2, and 3 weeks after adding water and or phosphorus to really see how this changed the microbial and viral community. So there was a lot of stuff that I did here. And so this is kind of when I wanted to get more in the bioinformatics to make sure that I'm feeding this crowd here that's these bioinformatics majors. So first we have this metagenome, right? And we've talked about this is the classic way we look at viruses. This largely captures double-stranded, excuse me, sorry, give me a second. You get these double-stranded DNA viruses. You also get microbes, right? So metagenomes are nice because you get a lot of information. And the downside is you get low resolution on viruses. So there's other things we want to do. Well, one way is kind of a virome. And this is what I largely pushed and optimized when I was a PhD student, and I use to this day. And this is where we physically separate the viruses. And I kind of talked about it before, but I kind of wanted to lay it out a little bit more so you understood that we tailor it to the soil. So I had to change it up now that we're working in grasslands. And we kind of had to desorb the viruses from the soil surface, whether there's more minerals, whether there's more organics, it varies. So you have to really change it up. You size filter them, and then you purify things of virus size and concentrate them. And then you extract the DNA from here. And so a lot of the people that have started using viromes have seen that this actually captures more viral diversity. We get way more viruses from this way. So this is a great way to look at viruses. A downside is you don't get the host information because you remove that, except for if it's ultra small bacteria or archaea, because then you will get that. So that is a way to concentrate and look at them as well. So I did both of these methods on my soil samples. But then in addition to that, I also did a metatranscriptome because I wanted to look at all the viruses. So with the metagenome, I got a large bulk of our double-stranded DNA viruses in and out of hosts. The virome was large, or the virus is largely outside of the host, and this can be single and double-stranded. And the metatranscriptome let us look at the RNA viruses, right? So we can capture RNA viruses, and we can actually get gene expression to pair with our stable isotope probing information for our hosts. But the problem is, if you've ever worked with a metatranscriptome RNA, most of it's rRNA. So you have to deplete that. And who do you deplete for? Can you deplete for everyone? There are some universal kits now, but you lose a lot of your information. And most of the information that we want is actually the mRNA because that's where the RNA viruses are. And then in addition to this, there are papers talking about how DNA and RNA can just exist in the soil matrix, and it can be called extracellular DNA, it can be called relic DNA, it can be called ancient DNA. So I thought, okay, well, why not try to see if I get anything from here as well? So then I just desorbed DNA from the soil by washing it, and then I size filtered it to a 0.02 micron filter just to allow DNA through. So not even viruses got through. And then I sequenced this just to see, okay, what's the background signal? Because DNA is important because it's a nutrient that things can eat, part of biofilms, but it also can inflate our richness if we're doing a metagenome. So I use this to help remove noise and characterize organic matter. With all of this, this was a ton of work. And I realized that I'm not going to be able to talk about all this, but I wanted to put it out there because we love taking students. And if any part of that excited you, reach out to me to come for the summer, or there's a lot of fellowships I'll talk about at the end that you can do during the school year as well. But I don't want to leave you hanging, so I will talk about one part of this project. And since I've been talking largely about DNA viruses, I want to emphasize RNA viruses. So we largely know nothing about RNA viruses in soils. There's a handful of papers, and we really are seeing that they infect bacteria and archaea, but then they also seem to infect fungi and these other eukaryotes. So maybe we need to be studying RNA viruses just as much as we're studying DNA viruses. So if you remember back to the study, I added water and then I added phosphorus to the study. So I wanted to see, okay, how is this affecting the RNA viruses? So first I want to draw your attention to the right side. We have an ordination plot. So the axes don't really mean much except for really how close points are together is how similar they are, and how much further they are apart is more dissimilar they are. And so the axes kind of guide you on how much of that variance is being explained. So what you should, and sorry, this is the overall looking at the viral communities. This is a way of looking at beta diversity. And so here, each dot is a different sample that I had, and I had triplicates. So the first thing you should see is in dark brown, the circles. These are our dry soils that I extracted viruses from. You'll see that those are clustering together and far away from the other samples. This means that our dry soils have a different viral community for RNA viruses than our wet soils. So wet up alone changes our RNA viral communities. Okay, so we see that. The next group you'll see in the middle there are our communities 1, 2, and 3 weeks after adding water, and I did not add phosphorus. So those cluster somewhat together, but they're different than the dry ones. But then they're also different from the ones to the right, which is the same communities that I added phosphorus to. So we know adding water changes the community. Adding phosphorus also changes the community. So this means that we know that phosphorus is affecting these RNA viruses in some way. So that was looking at the beta diversity. Now on the left side of the plot here, we're kind of looking at the alpha diversity now. So at top, we have our Shannon's diversity, and this can be broken down into a species number or richness, which is the second box plot. And at the bottom, we have our evenness. So for richness, you're looking at the number of unique viruses in the sample. And then for evenness, you're looking at overall, are these viruses have the same abundance, or some more abundant and some are less abundant? So looking at these three together, we can try to understand these communities more. So if we look at the top one, what we see is, in the light blue, when you add phosphorus, the diversity decreases, and that's significant. And that's interesting. We know that it changed the community, and my hypothesis was if they're limiting phosphorus, I thought maybe by adding phosphorus, it would increase infection, but we're seeing that diversity decreased. So this is why it's important to tease apart what goes into diversity metrics. So what goes into Shannon's diversity is the richness and the evenness. So if we look at the richness, the species number, we see, oh, that didn't really change. So while diversity changed, the number of unique viruses, that didn't change. Okay, so if we go to our evenness, okay, now here we see a significant difference. In our dry soils and our undermined soils, they're pretty much the same. This means that there's a lot of viruses and they're all about the same abundance. But then we see that when we add the phosphorus, that there's a range. And this shows me that disproportionately some viruses are benefiting from phosphorus and some are not, because some are more abundant and some did not increase in abundance. So it's caused a schism in the community where some are benefiting. So we wanted to see, okay, we know that water and phosphorus is affecting our communities. We know that it's actually only benefiting some of our RNA virus community. So who is it benefiting? So looking more into the taxonomy, we have two figures here looking at the cumulative abundance of viruses based on who they're predicted to infect over time. So what you should see is on the left side is we have the phosphorus soil, and on the right we have no phosphorus addition. So on the right, you can see, okay, the blue line's a little bit above, but it's not significant. On the left side, the blue line is high above, and it is significant. And so what we see here is based on these viruses linked to their hosts, all the different hosts, that only the viruses that infect bacteria, so the bacteriophage, are benefiting from adding phosphorus from the soil. So my large hypothesis here is that in soils, we have these invertebrates, plants, we have fungi, and if they're phosphorus limited, they have roots, they have mycelia, or they can move, and they're going around trying to scavenge phosphorus. Where if you're a bacterium, you're kind of just stuck in these soils, and it's really hard to scavenge these phosphorus. So when phosphorus is added, the bacterium does better, and the viruses that infect the bacteria do better as well. So by adding phosphorus, we are increasing viral infection at bacteria in these soils. So it's just kind of a better understanding of viral roles and phosphorus cycling there. Okay, So I don't know how well I'm doing on time. I have one last slide, and I threw this in just to kind of outreach to students who could be interested. So the
You're doing
national,
fine on time, by the way.
oh, okay, great, good, good. Um, so really, if there's anything you're interested in that I talked about today, or even other aspects of the project, maybe you don't care about viruses, just reach out to us because the national labs have a lot of ways for students to come work or work with people in national labs. So in blue here I have kind of highlighting just students. Doesn't have to be just during the summer. We have these academic cooperation programs. So these are for people, let's say you're not in school anymore, you're leaving. You have a bachelor's, your post-bachelorette, or you finished with a master's. You can come work in a national lab for 1 to 2 years. And the purpose is you don't stay there indefinitely. You get research experience, so you can either go back to grad school and get a PhD, or you go into academia, you go and get a job, right? So it's really to give you research experience. You can come in as technician. Um, sorry, the academic graduate appointee is what I was talking about. The academic cooperation program is if you are a current student and you come work at the lab. So we have different programs whether you're currently a student or you're not. And then if you are in the military as well, we have special programs for that. There's also larger DOE ones, so maybe you don't want to work at Lawrence Livermore National Lab. There's other national labs you can work at. And so the ones that I've highlighted here, there's a lot for graduate students and there's less for undergrads, but the Soli one is a great one where you can come work at a national lab for 10 weeks. They have special ones for students if you know they're at community colleges, but most of you are on the California state schools. So you can take advantage of the SLE program. There's also graduate programs too. So if you want, I have links there for the other DOE programs to come work at a national lab. So a lot of these are funded and they're funded for 10 weeks up to a year or two to come work with someone at a national lab, either to enrich your current work or just to get experience. Okay, before I end, I just want to acknowledge different funding agencies, and I want to do a land acknowledgement for the areas that I worked on. And then, so with that, I will take any questions.
This was awesome. Thank you.
Great talk.
Yeah.
I'm sure some minds were blown. Trillions of cows, just the visual of just trillions of cows must be really kind of amazing. Yeah, that was really great. So we have questions, a lot of questions actually, really good ones from the students, which I can read out. There was one in the Q&A, and then I have a whole bunch I can read. So we can start with the Q&A, which is, what is the best method to lyse bacteriophages?
Oh gosh, that is a hard question. So bacteriophage are not created equal. They persist differently. They infect different hosts. Yeah, there's not going to be a great answer, but I would say you largely have to figure it out for what you're looking at. The gold standard, which is in the literature the most, is mitomycin C. It was this cancer drug. Somebody saw a long time ago it could induce phage, so it's largely used as a standard. There's been numerous papers, and I've done work myself, where we show that it's actually not the best way. All right, so it selects for some and others doesn't select for. Um, if we think about, I think the reason why it's hard to answer that question is we don't even know why viruses want to be induced, right? If they're temperate viruses, then they can either remain in the host or they can switch to lytic and kill their host. But what causes a switch? We don't know. It used to be they're stressed out, they're leaving. But we've also seen that when they're happy, that they also want to stay in their host, or they also could want to leave. There's also been work showing that viruses talk to each other. They can co-opt the quorum sensing of the host. They can cleave peptides or add peptides on and communicate with other viruses to guide these kind of lysis-lysogeny decisions. I've seen in soils that we've added succinate, and that worked really well. Even adding water, you saw from my work, by adding water, it changed the community. We have a lot of viruses that will lyse. So I think the best way to lyse viruses is just to cause maybe some kind of disturbance. But I don't even know
So
if that's
sorry, it sounds like you started two different issues there, because it sounds like there's bacteria that are or the viruses that lyse the actual bacteria. But
Yep.
there's also the issue of, if you have viral particles, how do you break them open I think there's a lysis for that, right?
Oh, I see. Yeah. So
Yeah, so
that's
those are two
right.
different things, but
Yeah,
you're talking about blowing out the bacteria, but lysing
yep.
bacteriophage is a
Oh, sorry. I I misheard the question.
no,
I
it's
see what you
okay.
mean.
Those
Talking
are
about,
both great. I want to hear them both.
yeah, you're talking about breaking open a
Yeah.
virion, right?
Yeah.
So for that, there's been many ways to do this, and a lot of the standard kits work. It's basically a protein complex. So Protease K, these enzymes that can just break them open, that works well. I've used heat. Heat works really well to denature these protein complexes and make them open up. The problem is you want to break it open without destroying nucleic acid inside. So there's always a balance of what you want to do.
Here's some questions. Can metagenetics distinguish between bacteriophages and phages are integrated into the bacterial genome? And if not, does this skew the metagenomic data?
Yeah, absolutely. So we can work towards this. So this also depends on how good our methods are for assembling bacterial genomes, right? So when we do a metagenome assembled genome, we kind of bin these contigs together and we do a refinement. So arguably, if you have high confidence, like a high quality mag, that all the contigs in there are genuinely supposed to be there, then that makes it a little bit easier. Because what we can look for is we can look for a virus to see if it's integrated or if it is one of those contigs and it wasn't just misbinned, right? So if we see it in the genome that way, we know, okay, it's in the host. So that's a great way. But binning is still not perfect. I think we're still far off, but we're working with what we can. And then if you have something that doesn't bin, you could arguably say it's not in the host. Or if you take it and you break it from a virion, therefore you know it's not in the host because it came from a virus particle.
So if you had both, you could say, well, at least we know these ones are free. But you wouldn't say that everything else, like if you did a metagenomics and a free virion thing, you could at
Yeah.
least say which fraction were not, but you couldn't say, oh, these are incorporated, because there might
Yeah.
be a fraction that you just didn't get out,
Yeah.
especially in
And
soils.
there's actually other parts too. So I have a 2018 paper talking about the under characterized roles of viruses in soil ecosystems, where I compared our metagenomics to our viromic approach and really have a figure laying out kind of differences. And it could be that in a metagenome, what I think so far is that we largely have seen viruses that are integrated in the most abundant host. So we're skewed to the bacteria that are most abundant. So we're seeing those viruses, but we could be catching viruses that are integrated or ones that are actively infecting at that snapshot. So you have to remember a metagenome is a snapshot in time. So they could be lysing their host at that exact time and we happen to catch them. You may also get a couple virions that break open as well too. So I think metagenomes take a little bit of that, and that's why we see some overlap. And the virome is just everything that's created a virus capsid, which for DNA viruses, we think that's all viruses, but that's not the same for RNA viruses.
Now, if you did see, this is one thing I wondered, if you do a lot of sequencing and you sequence and can do assemblies, then you can potentially tell that there's a bacterial gene right next to a viral gene. And then you were like, oh, that must be incorporated. Is that something people do?
Uh, yeah, so you can look for flanking of genes, right? Um, so that's part of it. Whenever you look at viruses, you have to remove contamination. There's always a lot of host contamination. You could arguably say that if you have host genes on both sides of the virus genome, that it's integrated, right? Because if you see it on one, that could be just the virus took some host DNA with it. And did you actually look at where the virus genome started and stopped? Do you know specifically if that's correct, right? So there's a lot of things to think about. But if it's flanked on both sides, you could arguably say that it's integrated. There are
Well, if you had
differences.
really long reads, you could probably do it, but
Long
yeah,
reads definitely would answer
yeah.
that. And I really like the idea of long reads because you get these hypervariable regions that are really hard to assemble, right? You get these genomic islands, which viruses can be in or and contain as well. And then you can actually get whole virus sequences. So long reads could be very helpful.
Yeah,
Yeah,
that's
and
great.
as Yumari correctly mentioned, right, if you have things like horizontal gene transfer, that messes that idea up, right? Because for all you know, it's actually just actually an AGT event and not necessarily mapped to the actual virus. So
Yep.
anyway,
And you have to think that if a small portion of viruses contain AMGs from what we see, AMGs are host derived, so they look like host contamination. So if you see them at the end of a genome, then you're like, oh, maybe it's not an AMG, maybe it's just contamination. So there's like methods showing that not only if you find an AMG, but you have to try to find it in the middle of the genome as well to make sure it's not a contamination event.
Cool. Another more broad question was what could it why is there so much carbon in the equator and the Arctic? What is your answer for that? Is that historical? What is that?
Yeah, so along the equator, you have high production, high activity. So you have a lot of biomass. So there's a separate figure I probably should have showed, which shows where carbon is in the terrestrial ecosystem, whether it's in biomass or whether it's below ground in soil. And if you look towards the Arctic and the equator, you have a lot that's above ground and below ground, and it can vary. So along the equator, you have these tropical rainforests and stuff. So you have a lot of biomass above, right? And then the biomass falls, and then you get a lot of carbon stored in the soil. Just because you have a lot of carbon being produced, it's highly productive, right? But in the Arctic, it is different. We have a lot of carbon stored in the Arctic just because degradation is limited due to temperature and other artifacts. Like in a bog, you have the sphagnum moss, it produces these solutes, and the solutes, an example would be galactochloric acid, limits microbial growth, right? So the microbes can't break down carbon as much because they're limited. Also, the temperature limits them. So if you've ever been up towards the Arctic, it's not in all areas, but in Sweden's a great example. You get these peatlands, and you get this peat, which is just a ton of undigested organic matter, this plant material just laying on the soils. And when I take peat soil, it's largely just plant material and barely any soil. And I swear that's where they got the Tempur-Pedic Swedish bed from, because when you walk across it, it's exactly the same, where it's just you're stepping and pushing down all of this plant material. And it's just
Springs
this is
growth.
slowly yeah, it's just slowly degraded. So you just have this accumulation of carbon over time.
Amazing. Yeah. No, I was going to ask if you, and this sounds maybe bad idea or crazy, but I understand that trees are also moving north into these areas. Is that right? You're getting more tree as it's a warming climate. Are you getting any invasion
So
of trees and other plant life?
yeah, so it's not my main area, so I will say what I know. But I know with climate change, we are getting longer warming seasons. And, for things that can move, like animals and stuff, they're moving where their habitats are. So as it's getting warmer, things are going to be moving more north or more south to get to conditions that are favorable. Trees can only move so fast, but it does seem like they're moving. And I'll also say that in the Arctic, there's a lot of these rich soils. So as temperatures increase, these are highly fertile soils. So there's issues that are we going to use them for agricultural reasons? That wouldn't be so good. But also, if you're thinking if you're a plant, when those areas they have conditions that are favorable for you, like temperatures and stuff are fine. There's a lot of nutrients. You would want to be up there. So I could definitely see how we're going to have new crops growing because conditions become favorable.
Would
Or
it be
not crops. I want
would
to
it
say
be
plants.
would a geoengineering solution? I know it's a bad idea, but if they've got that much methane burping out, this is a massive, massive methane bubble across the entire Arctic, which includes all of Siberia. Would it make sense to go
Yeah.
plant trees? I mean, do you know if that would actually change how much methane and carbon dioxide is used? Like, I know it
There's a,
sounds like a terrible idea, but maybe you
there's
should
a
just fricking plant trees.
yeah, there's a lot to
They're
unpack
going to be
there.
there anyway, right?
I will okay. I will first say that I don't think geoengineering is a bad idea. I think we need to explore all of our options, and any way that we can capture carbon and store it is important. And LML actually has a roads to removal publication that came out recently showing ways how different parts of the US and stuff can store carbon. And we actually focus on grasslands rather than trees to store carbon, because as a tree, you store some carbon below ground, but you can physically see a tree. The carbon's above ground, which means it's susceptible to changes in climate and drought, fires, where grasses store the carbon below ground, especially if you see switchgrass, these meter long stretches of roots, where if the top burns, you still have the roots below ground, so they're stable, right? So you have carbon storing below ground. So even if we theoretically planted trees in the Arctic, that if the trees grow, that's great. It's storing it. But how reliable is that? What if a big fire comes in like we're seeing in Canada and stuff and just wipes them out, right? So I'm just indirectly avoiding answering the question and saying that I don't think the solution is to cause change because we see that every time we try to change an ecosystem, we make it worse. I would say that there's different ways to manage it. Like we think of wetlands releasing methane, but we actually see that highly productive and high quality wetlands can actually help store a lot of carbon and protect us from flooding. So the solution should really be to keep the Arctic cold.
Good luck with that one. Okay.
Yeah.
You're actively heating it up. So let's see, some other great question. Let's see.
There's one in the chat. So,
Oh yeah, go for that one.
I like this one. Yeah. So it's, the question was, since metagenomics is sort of like a snapshot in time, shouldn't there be a bigger push of co-assembly to capture more over multiple samples or multiple time points?
So it's not even just co-assembly, but you're exactly right. Getting a temporal and spatial component to it. Absolutely. At some point, we're going to take so many metagenomes, the Earth is going to be Swiss cheese, though. So there's also a push that maybe we need to sample to get databases up, but at some point we need to start looking from genome to phenome and really predicting change, especially with climate changing, and modeling this so we don't have to sample and we're not just lagging behind, that we can anticipate change. So I think one thing is, yes, temporal sampling is important to see it changes over time. But, I would argue using activity measurements, whether it's metatranscriptomes, whether it's bioorthogonal or non-canonical amino acid tagging, like different ways to look at expression or stable isotopy. So that way, instead of just seeing everything that's there, you can look at who's utilizing what substrate, who's active at what points, right? And that way we can get a better understanding and feed that into our models.
This is a really good question. It comes up a lot, and it's great to explain it to people because so many people are confused. Is there a structure analogous to the bacterial 16S ribosomal RNA that can use like a marker gene to characterize soils? I know you know the answer to this, but this comes up. You must hear this all the time too. Is there's not a like explain about the marker gene thing, like so,
Yeah.
and what you can and can't do.
So, marker genes came up originally because sequencing was really hard and took a long time. And it would be nice if you could just sequence a small portion of something to really understand what's going on there. Now that we have metagenomes, we can actually get more beyond a marker gene and understand the rest of the genome and their capabilities. So the problem with the marker gene, I'll first off saying, is it just gives you taxonomy. Now, there are marker genes for bacteria. There's a marker gene for archaea, for eukaryotes. For viruses, though, I think that's what the question is, right? Soil viruses. There's not a marker gene for viruses, right? We have different viruses can have different subunits on their capsids. So you can look for marker genes for specific capsids. A lot of single-stranded DNA viruses will have some marker genes for how they replicate. For RNA viruses, there's the RDRP, which is, oh gosh, I always forget it when I go to say it.
The
RNA-dependent reverse
polymerase,
yes.
Something like that, yeah.
So this is a gene that's specific to RNA viruses that's not in microbes. So we largely use that as a marker gene for RNA viruses, but not all RNA viruses have one. So we can't just say by using that, we're looking at all RNA viruses. Yeah, so
you basically, there's no single marker gene that for all viruses. They don't have a ribosome. They don't have it. You could look at groups though. This is really good. I would say I'm going to push back on the metagenomics can do everything because for two reasons. First, I do metagenomics, and you absolutely have to have a database to know anything. So if you sequence a bunch of random DNA and you don't know the first viral work, they didn't know anything because they had no viruses. Whereas a 16S, it can only tell you taxonomy, but because for bacteria at least, because they all have it, they can at least put it in a tree somewhere and a phylogeny and know it. And that's still useful because there's still a lot we don't know. I agree. And the other thing is you have to be careful with like large organisms like fungi. And I'm telling this to other people because they have giant genomes, right? And so we still use a marker gene for the fungi, which you didn't talk about at all, which must be a very important player in soils. But you can't, there's databases for fungi are almost non-existent because you have to sequence entire 3 billion base pair genomes or 10 billion to get a fungi. Maybe it's less than that, but it's huge. And so I'm still thinking but the viruses, absolutely 100%. You can't know anything pretty much for a lot of stuff without metagenomics. So, but all the metagenomics people are like, oh, it does everything. It's like, yeah, hang on a second.
I don't know. Okay, I don't know if I said that. Because I don't think metagenomics can do everything, and I will push back on that as well. I'll say that I didn't talk about fungi for a couple of reasons, but the first thing I want to say is exactly to your point. We can't assemble metagenome assemble genomes for fungi like we can with bacteria. They're really big. There's a lot about their genome that's different. So we definitely use marker genes. Specifically, we'll use like ITS, 18S to look for their genome or look for, marker genes for them, right? So that is a big thing. For viruses specifically, it appears that most double-stranded DNA viruses infect bacteria in environmental ecosystems. We don't largely see fungi. And most of what we do see from fungi have come from isolates. And what we think so far, based on what we've seen, is that most fungi most fungal viruses, which we call mycoviruses, have double-stranded DNA or double-stranded RNA genomes. And a lot of them don't have a virion stage in which they have a capsid. There'll be these strands of RNA in the cytoplasm. But that's just what we know from what we've studied so far. So we don't know if that's the truth. But from the work that I've done, when I do DNA work, I don't see fungal viruses. When I do RNA work like the one I showed you, we see fungal viruses.
Very cool. This is really cool stuff. Fungal viruses are just a whole nother level of mind-blowing stuff. But fungi are so interesting too, because I studied them as well. I studied most in the built environment. So I've studied all three parts of life. And the fungi are really annoying because they're the same cell over and over and over and over and over and over again. So it's really like you have to kind of figure out the biomass of them to actually know their relative abundance. Whereas with the virus, it's a single particle. With the bacteria, it's a single cell, right? So if you count them, you're like, okay, I can relate it to abundance more or less with the sequence. You can't do that with fungi because you get like one thing. I mean, it's really crazy. I'm gonna ask one more question. I thought it was good. And then if you don't mind, I might send you the link to all these other great questions. If you have time and you want to look at them,
sure.
You can put a little side answer if you want. It's up to you, but there's so many good questions. But I just want to ask about the national labs. How competitive are the internship programs, and what kind of things would you highlight when you're applying? Do you have any ideas on what you should do?
I would say they're competitive. It also depends on what fellowship or application you're applying for. So we have some that are much more competitive where they fully fund you for many years and they give you funds to do your own research and stuff. And then we have the more general ones, like I'm a research scientist, you want to work on a project, you reach out to me, I fund you. Because then it's less competitive in the sense that you're not competing across the whole lab. You are just reaching out to me. And the competition depends on your willingness to do work. So every summer I'll give talks like this. We do a lot with people from Georgetown. We have a scientist that used to work at Georgetown with us. So we have a lot of interaction with summer students from Georgetown, especially master's students. Where I'll give a talk, they'll reach out to me. I'll have a couple CVs, I'll go through them. But the students that have the most success are the ones that reach out to me and tell me what they want to work on and what they liked. Because then I don't have to look through a bunch of CVs. I see that you're interested, you're motivated, you sent me your information, I can zone in on you. And if I have funds at the time, you're who I hire. So what I look for is somebody who is willing to try, right? It's going to be hard whether you know bioinformatics or whether you know what lab. Just be willing to whatever you work for or whatever you want to learn, just let me know. Because I always like where when you do something, you don't want to just do something you've already done. You want to learn something new. So you kind of tell me kind of what you know and what you want to learn with no false pretenses that you can do it all. And then we work together on that. And for a lot of the virus work, it's virus specific. So backgrounds with metagenomics and stuff is helpful, but a lot of the scripts and stuff is specific that you have to be trained on. And virus knowledge is a lot of different. It's different than regular microbial ecology. So a lot of people I have coming in are brand new to it, and that's okay. So I look for somebody who is willing to work with others, not toxic. I don't like the person where when you jump on a call and they're constantly just bad mouthing others, right? We are humans and this is a working environment. So someone who's easy to get along with, somebody who wants to try, and somebody who's interested in working. That's really what I look for.
Yeah, we train our students in, well, I do bioinformatics with Python in the fall, and I assume Python's a very useful language a
Yeah,
lot of the
I'd say
time.
Python's the main language I
Yeah.
use, and then R.
Yeah, this is great. Do you hear this, people? I teach this. This is why. Bioinformatics, Python. And then we do Unix, R, and object-oriented Python in the spring. And then in the spring, we also do metagenomics and RNA transcriptomics. And we do, like we go through, we're actually going to get into metabolomics. We do have some
Nice.
virus examples and genome assembly. We teach about, hidden markup models and all this kind of stuff. So, and then we do QIIME. But we also do assembly, we just do a smorgasbord because we know you're not going to be able to do every single program that's out there. But
Yep.
we want to know the fundamentals, but we also want you to be able to be flexible. So you know something of these things. You can do the programming languages. So when you get to a place, you can say, I can learn the new scripts. I can learn how to
Yep.
install something. I can learn how it works. And that's what I think an advisor would really want to know, is that you're eager to learn new things and you're willing to troubleshoot and try, and you already have a baseline that gives you confidence that you can actually do these things. So yeah, so it's
Yep.
impossible to know everything, but it's possible to be a person who can learn things quickly.
Absolutely. So I'll say that a typical student that I get that I really like is they have experience working in an HPC environment. They're quite good with bash commands. If they know some Python, that's great. Some R as well. I think R Studio to make figures is a quick and easy thing to do. You can do pandas and Jupyter Notebook, and that's great. But I would say bash command is the most important thing. Having some Python is great. Having some R is great. But really background on how metagenomics works, how assembly works, what the errors behind assembly, what are things that you have to think about. I think that is the most important thing, really understanding what methods we use, why we use them, what are the pros and cons associated with it, because then we can build from there.
Sounds good. I do De Bruijn graphs and all that kind of stuff. So
I exclusively use those. So
yeah, exactly. Like spades, meta hit, that kind of thing. So yeah, no, that's fantastic. It's good. It's good for me to hear that my class is actually useful too. So yeah, that's great. Well, people definitely might reach out to you, if nothing else for advice or just all that kind of stuff, but maybe to work with you. And I just want to point out everybody that Maya Breitbart, who you cited several times, was a PhD graduate in our program, and she actually worked in a joint lab space with me. So I'm on some of those some papers with her, but she's now in Florida.
Florida.
And
Yep. She's fantastic.
yeah, she's also someone who probably would like some of our students to talk to and work with. She might have money,
Yeah.
but these national lab jobs are amazing.
The last thing I'll say that if you're interested in doing work for the summer, I have a lot of students that will wait till May to reach out to me, and then you've missed the ball if you wait till May. We typically start looking for summer students in February and finish by March. I have taken
That's
students
really
that
good for
reached out
them.
at the end of spring, but usually you have to have your own funding because any funding I have, I've usually already allocated because it takes time to do background checks. I start meeting with people weeks beforehand, giving them papers to read to get an understanding and make sure that we both develop the project together. That way, when you come, we hit the ground running.
Fantastic. I'm glad to have this all on record too, so people can look it up. I will try to remind people in my class, hey, this is a good time to reach out to Gary and reach out to the national labs because it's too late. If you wait too long, it's too late. But you want to get enough that you're like ready to go and competitive at the same time. So
Yep.
fantastic talk. I really loved it, and I know a lot of people did. So good luck with your research. You're doing a very hard task, getting viruses out of the soils. And what I think is cool is you're coming up with new technologies, essentially. So you're almost like tech development because you're just trying to solve an awesome biology problem. You just have to
Yep.
come up with new solutions and new things all the time. And that's just what science should be all about. So, all right. Thanks so
Thank
much,
you.
Gary. What a pleasure. And we might reach out to you about our soil stuff that we're doing at some point. So I'm really glad to talk to you about this.
Of course.
Thanks for presenting.
Okay. Take care.
All right. Take care.